I'd like to talk to you today about a project I've been working on since the early days of the Glycan project called GlycoMotif. GlycoMotif is a database and website of glycan motifs and determinants, along with their alignments to all of the glycan structures in Glycan. You can find GlycoMotif at glycomotif.glyomics.org. This is work with a person in my lab, Wenjin Zhang, who's going on to do a PhD next year, and Renee, Mike, and Will from the Complex Carbohydrate Research Center. GlycoMotif is an organized, curated aggregation of glycan motifs from various different glycoinformatics resources, including Glycan, Unicarb KB, glycoepitope, and the CCRC. These glycans have been organized and curated. They are comprehensively aligned against all of the structures in Glycan. And in fact, we don't just do one type of alignment here. We align the glycans depending on their semantics at various positions in the glycan, either at the core, at the non-reducing end, internally as a substructure, or to represent the entire glycan. These alignments are available for interactive browsing or download. We curate semantic names, keywords, and references for each of these motifs, and we encourage folks who are interested to contact us because we need help with this. The Glycan collection is based initially on the Cummings 2009 paper and has been integrated with Glygen, but has undergone a variety of curation and cleanup exercises since. In addition, the Glycan collection forms the basis of Glygen's glycan classification algorithms for glycan type and subtype. You can see a screenshot of the homepage down below. In this slide, you can see 4 different views of the information available in GlycoMotif, a list of the keywords that are currently annotated as part of the Glygen motif set. These include things like lactinac, various different blood groups, and a variety of other types of glycan characteristics. Again, we encourage folks to contribute keywords and categories that can help us understand these motifs. Here's a Glycan detail page showing names, keywords, publications, and the various alignment information. And below on the same page, we have information about all of the Glycan alignments that align, Glycan structures that align to this particular motif, including whether they are aligned strictly or non-strictly, respecting the annotated positions exactly or not. And then lastly, everything in GlycoMotif is available for download, and you can access it using the Sparkle endpoint. A number of specific queries are provided for you to make it easy to download and access the information in whatever form you find useful. A new feature that we've added to GlycoMotif recently is the ability to align novel glycans that are not part of GlycoMotif to the to all of the motif structures in GlycoMotif. You specify your GlycoCT or Works glycan sequence, and the motif match web service aligns that structure against all of the GlycoMotif entries. You can use this web service directly, issuing a web request and getting back JSON format information. You could even run your own copy of this using Docker on your own computers so that you can query it directly without waiting for my server, or you can use it via the GlycoMotif website. When you enter your sequence, it is sent to the web service, and what comes back is information on the specific structure you requested, and images constructed on the fly. And down below, you can see that we have constructed or we found all of the motif matches within the Glygen collection that match to that specific structure. The structure may be completely novel, never seen before, or it could be one that exists in Glycan. This infrastructure doesn't care. All right, well, that's all I have time for. I encourage you to go check out the glycomotif.glyomics.org website.